A very good afternoon, ladies and gentlemen. I, Nitikesh Prakash, welcome you all for the next session of the Research Scholar Day 2021, the annual research festival of IIT Madras. So it gave me great pleasure and honor to welcome our speaker for the session, Dr. Upinder Singh Bhalla. Thank you, sir, for joining with us. Dr. Bhalla is known for his study on neuronal and synaptic signaling in memory and olfactory coding using computation and experimental methods. He is an computational neuroscientist and professor at the National Center for Biological Science of the Tata Institute of Fundamental Research. He is an elected fellow of the Indian Academy of Science and the Indian National Science Academy. Dr. Bhalla has also been conferred with the prestigious Santi Swarup Bhatnagar Prize for Science and Technology by CSIR in the year 2007. The title of his talk is Learning in Artificial, Real and Social Brain. Over to you, sir, and welcome on board.
Thank you very much, Nitikesh, and thank you to all of the organizers. It's really a pleasure to come and speak to at the Research Scholars Day. And I've often visited IIT Madras, of course, and it's unfortunately one cannot do so in person in these times, but I hope to do so again sometime soon. So what I'll do is I'll just shift over and share my screen. And please let me know if this is coming through properly. Are you able to see it?
Yes, sir.
Okay, very good. Okay, so I'll go ahead and I don't know quite how you organize the questions. Will they be in one group at the end or can they come as we go along?
Sir, we are live on the YouTube. So,
okay.
So in between we can have a question or at the end of the session, mostly we'll have.
Okay, maybe we'll have it at the end.
Okay.
Okay. So anyway, as I said, it's a great pleasure to be virtually with you at the Research Scholars Day. So I'm going to talk to you about learning. Learning is my passion, or at least studying learning is my passion, and doing learning is also my passion. And I'm going to try and stitch together perspectives on artificial brains as an AI, which you're all very, very familiar with in this day and age. I'll focus mostly on real brains, though my approach to studying them is, as you will see, very substantially computational and mathematical, though we do a lot of experiments as well. And I'll end with a little glimpse of the whole point of these brains, which is to function in a social environment. So without further ado, let me get started. So this is going to be the structure of the talk. I'll start out telling you a little bit about some metaphors for the brain, and I'll sketch out how learning happens. Then in the main body of the talk, I'll talk about key operations that happen when one learns something, what to learn, when to learn it, how to store information, and how to retrieve it. And then I'll, as I said, end with a little perspective on curiosity, learning, and survival. So let's start with metaphors. So I always thought this was a great quote, which basically says that the metaphor used for the brain tends to be that of the dominant technology, of the most advanced technology of the day. And this is how people have, over the ages, thought about how the brain works. And here are some examples, right? Long ago, in the times of the Greeks actually, I don't know if you were aware of this, they had some very elaborate capabilities for hydraulic devices. And even to this day, many of the terms that they came up with this hydraulic metaphor are still used when talking about what our brain functions. You know, you say someone is bilious, in other words, they're in a bad mood, or they're phlegmatic, they're sort of slow and not very energetic, or sanguine, optimistic. Sanguine means bloody. Yeah, blood is supposed to be a source of energy. So I guess that's where that one comes from. So these are hydraulic metaphors, and they are ancient, and they're very much in today's language even now. There are mechanical metaphors. You may be familiar with this, right? There's, of course, this is a simple watch. Here's a much more complicated thing, which is an implementation of Babbage's analytic engine. So these are mechanical metaphors for the computations that the brain might do. Though I don't know if you noticed, I slipped in another metaphor there. I'll come to that in a moment. So the idea of that these metaphors is that maybe this is how the brain works through cogwheels and levers and so on. This is a very beautiful metaphor. It's from this quote from Charles Sherrington, who says, "Swiftly the brain becomes an enchanted loom where millions of flashing shuttles weave a dissolving pattern, always a meaningful pattern, though never an abiding one." And so this is how he envisions the functioning of the brain. And there's a gorgeous tapestry, which I think is an appropriate illustration for the enchanted loom. And if you look very closely at the tapestry, remarkably, it looks like the connections to some kind of a circuit. So yeah, many layers of metaphors, if you like, in this picture. Here's one of the somewhat more modern metaphors, that of the telephone network, which in maybe 100 years ago was handled in this manner, where people, in those days almost entirely women, were employed to plug in connections from caller A to caller B. And that was the dominant technology of the day, or the advanced technology of the day. And that was the metaphor for the brain. Now, much more recently, we are familiar with the computer metaphor for the brain. And I won't belabor this. I'll just make a point on this, which is I personally think that the computer metaphor is not just a metaphor. It is actually a functional description. But maybe we can discuss that at a later point. And as you can see, beautiful, very elaborate connectivity. And so this is now considered another form of metaphor for how the brain works. This has been taken yet further through the development of what's called connectomics. And the result of connectomics, so what they do in connectomics is they slice up brains with exquisite, this is tens of nanometer precision in 3 dimensions, and do very elaborate tracing algorithms to establish what is connected to what. And this is a very big challenge because, as you can see, the brain is absolutely packed densely with connections and cells and synapses and so on. And that's just illustrated in these little reconstructions of just a small segment of the brain. So this is another metaphor. And now we come to I would say, the most recent metaphor, which is artificial intelligence. And I kind of find this an amusing metaphor because, of course, artificial intelligence actually is derived from ideas that came about many years ago about how brains function. So now we have a sort of inverse metaphor for how the brain works based on machines of our own creation, which were meant to imitate how the brain works. Kind of amusing that. But I just want to give you a bit of perspective. Yeah, you can see how monstrous, how massive this thing is. Yeah, it's a whole room. It's full of racks. It probably consumes a megawatt of power. And what it tries to do, and in some domains it does well, and in most domains it does very poorly. What it tries to do is done so much better by this little thing here, which is about 1.4 kilograms, and it consumes a grand total of roughly 20 watts. Yeah. So this is what we really want to understand. All these metaphors are just ways to try and get a little bit of a picture about how something like this does all the things that make us human beings. So let's get into a little bit more detail into how this might work, with a special emphasis on learning. So I'll start out with a sketch of how learning might happen. And this is a learning paradigm, a learning technique that you are probably all familiar with, a very classic experiment, now over 100 years old, by Ivan Pavlov, who was actually interested in studying digestion, by the way. So in doing so, he was monitoring the salivation of dogs. And being a good scientist, he noticed something very interesting, which is that in his experimental lab, when the dogs were going to be fed, there was a kind of an alarm to tell, a bell rang to tell the attendants that now is the time to feed the dogs. And he was monitoring their salivation. And as you all know, what happened was that after a few cycles of this, the dogs started to salivate at the time of the bell, even before the food appeared. Right? And then he went after this unexpected observation, the serendipitous observation, and actually produced a lot of very, very important data, which is in many ways the foundation for a lot of theories of learning. So the key thing here is the formation of an association. The animal has formed an association between the bell, which was previously not in any way linked to salivation. But the animal has learned this association, and this can be very well measured, and therefore it's an excellent tool for studying memory. So let's now try to see how one could do this using a kind of a cartoon dog. So this cartoon dog has 3 neurons, 3 brain cells. One brain cell responds to food. That's the important one, right? So it sees the food in the dish, and when it's active, this neuron, it causes the animal to salivate. Very, very straightforward. This is what's called the unconditioned response. The animal does not have to be trained to do this. There is, let's say there's another neuron which responds to a bell. And of course, initially, bell has no particular relevance to salivation. So initially, there's no effect there. And let's just, for the sake of completeness, you want to make this a good experiment, it has to have a control. You have another neuron that responds to light, let's say, for example. Okay, so now what do? We do the thing that Pavlov emphasized, which is you have an association. And we have it so that we have an association between the bell and the food, but between the light and the food, there's no particular association. So the bell comes, then the food comes many, many, many times. That repeats many times, but the light and the food are not in any way linked in time. So what will happen? Now let's just make as I said, a cartoon circuit here. So the bell neuron, let's say for the sake of argument, it initially has a weak synapse, a weak connection to the food neuron. So this isn't strong enough on its own. So the bell neuron can fire as much as it likes. It's not going to trigger the food neuron. But because of this association, this repeated linkage in time, slowly the strength of the bell neuron, and I've indicated by the letter here, B, it gets the strength of this connection. The strength of the synapse gets larger and larger and larger. And by strength, I simply mean how much effect does the bell have on the activity of this neuron. So as that gets larger, you end up with what I'm going to call a switched on synapse, and I'll come to details of that later. So the switched on synapse is now powerful enough, it has a large enough effect that now when the bell rings, the food neuron is triggered, and when the food neuron is triggered, salivation occurs. On the control side, the light neuron does not have this association in time, it's random. And so its synapse stays weak, and there's never any association formed. So even though you might have given the same number of stimuli for bell and light, the one that is learned is the one which is associated. So this is a cartoon dog, right? Dogs have a lot more than 3 neurons. They do much more complicated things, but this is a good basis for starting to study one of the simplest forms of learning, and one which is present all the way from flies and worms to human beings. So this is a cartoon way of thinking about it, and we'll come back to this illustration a couple of times. Okay, so let's move on. So now, where does memory happen in the brain? Well, the short answer is it happens all over the brain, but there's a particular part of the brain which is really important for short-term memory, and it's also important for learning certain kinds of information which need you to declare information. So like learned book information is needed. So a lot of this work was done on this unfortunate, well, at that time, young man by Henry Molaison, who had intractable epilepsy. And so the doctors went in and removed the site of the epilepsy, which happened to be this region called the hippocampus. And now you may have heard of hippocampus because, partly because of these experiments, it has become famous. But they removed the hippocampus of poor Henry Molaison. And what happened was that he could no longer form new memories. He had his old memories. He remembered, say, who had been president before his surgery. He remembered football games. He remembered all sorts of things, but he could not form any new memories. He could not even remember the doctor who came and saw him every day for most of the rest of his life. So this is obviously then a very key region to look for memory in the mammalian brain. And in fact, that's kind of the region in which I do a lot of my work. And many of the properties that you just saw in this toy example are easily replicated in this structure, and many other more interesting forms of learning also happen there. So now let's go through the brain. And I just want to give you a little bit more perspective about this incredibly complicated machine, complicated biological system. So here's the brain. As I said, it's about 1.4 kilos, about 1.5 liters in volume. It fits in there. Now, of this thing, if you zoom in and think of the size of one neuron, which is about the size of this little dot over here, I hope you can see it. Let's now zoom into that size. So here's a computer model of a neuron, roughly 500 microns in size, yeah, 0.5 millimeter. And so fine, so there's a lot of activity happening in the neuron at this scale. But now let's zoom in some more. If we zoom into a tiny piece of the dendrite over here, let's do that now. So this is 5 microns. This little cube of tissue is 5 microns. And the yellow structure here is what's called a dendrite. It's one of these branches. So one of the branches fits, a small piece of it fits in here. The branch itself is about a micron across. And now we're starting to get to the business end for a lot of computation, which is the synapse, the connection between two cells. And that's what these little red dots are. These are the connection inputs coming into this piece of dendrite from other cells. Yeah. So now let's zoom into the size of one of these red dots, the size of a synapse. So now we're going to go down another order of magnitude. And here what we have is 0.5 micron across from there to there. And now we're at the size of one synaptic terminal. Yeah, roughly 0.5 micron. And now you can just about start to see the molecules that actually do the work. So this is the presynaptic side. The postsynaptic side is just as complicated, maybe even more so. And you can see the molecules, you can see the vesicles, you can see the microtubules, the mechanical structures that give rise to it. You can even see the synaptic release site. Now, this is a reconstruction, but these are all actual protein structures, these little dots and tangles. So this is the size of proteins, and these are the molecular machines that do the computations that we'll be talking about, right? So we've gone over many orders of magnitude of size to get the size scale that I'm going to focus on, which is that of molecular events. So just to reiterate, the brain does computation at many levels. You cannot separate one from the other. It does computations at the levels of molecules, synapses, dendrites, neurons, circuits, whole areas of the brain, entire systems of the brain, like the visual system, and then you put it all together, you get the behavior of the brain. And these are intrexicably linked. For example, a mutation which will have a molecular effect will have very likely an effect on the behavior of the whole organism. The information flows back and forth. It is completely seamless, but it's important to keep in mind that you have computation occurring at all of these levels. So let's think a little bit about the concept of information flowing as chemistry. You're all used to the idea of information flowing as electrical signals. So think of chemistry as electrical signals, but with an identifier for each signal, because each distinct molecular species can convey a different kind of information. So it can be like a different wire all fitting together in these very, very tiny volumes. And another very interesting thing about chemical signaling as opposed to electrical signaling is that chemical signaling is very short range because the diffusion length constant is of the order of a micron. It's much smaller than even the size of a cell, whereas the electrical propagation distance in neurons is about 0.5 millimeter in many cases. So the size scale, the number of subcompartments you can have through chemical signaling is very large, and therefore you can do a huge amount of computation locally, a lot of parallel computation. And that's one of the reasons that the brain can do so much, so efficiently. So just again, to put it in perspective, here's a set of signaling pathways that I've been studying for 20 plus years. I'm not going to go into the details. This isn't the full reaction diagram by any means. This is just a block diagram, but this is only about 0.1% of the molecular network. This is what it looked like on my computer some 20 odd years ago. And the real thing is so much more vastly complicated, but just to realize there's a lot of stuff there to do computation. Yeah, just to reiterate, this is what it looks like physically if you could have a microscope sensitive enough to see all of these structures. And each of these little dots and tangles is a protein molecule or other chemical signal and can participate in these computations. And we'll see a little bit more about how these work. Let me just, as a final step in this intro, let me give you some numbers for the brain. So the human brain has roughly 10 to the 11 neurons. Each neuron has roughly 10 to the 3, 10 to the 4 synapses. So 10 to the 15 maybe synapses, 10 to the 14 or 15 synapses. That's a lot of connections. Yeah, we used to think it was a huge number, but now you actually in your computers, or certainly in your large servers, you actually have this kind of data capacity. Amazing what technology can do. In each synapse, you might have thousands, 10,000s of reactions in every single one of these synapses. The electrical events are fast for biology. They're of the order of a millisecond. Chemical events are somewhat slower, seconds to thousands of seconds. Structural changes take longer. But think of the longevity of these things. Yeah? The lifetime of a protein is actually not very long, days. But the lifetime of a brain cell has to be the lifetime of yourself, because the brain cells do not replicate except for very, very small, rare exceptions. Yeah? And therefore, these neurons, if you want to remember something and keep it with you for your life, you have to be able to store information for 100 years. So that's quite a challenge. And we'll see a little bit about that. Okay, so with that kind of sketch of the brain and how learning happens, Pavlov and all this kind of good stuff, let me get into some of the details, some of the meat of the system. How does learning happen? And there are 4 key operations I'm going to discuss: what to learn, when to learn it, how to store information, and how to get it out again. So what to learn? So we've already seen one kind of thing, which is associations. You need to learn when 2 things are related to each other, when they co-occur. Is there some causal link between them? Very often we infer causality by the repeated co-occurrence of events, right? So the dog presumably eventually thinks that the bell is causally related to the arrival of the food, which is a reasonable hypothesis to make. Yeah. So this is one form of learning. This is one kind of thing one must learn. Another kind of thing one must learn are patterns. I would say conjunctions of different kinds of stimuli, right? So if you're walking along, say, in a busy bazaar, you will have a conjunction of stimuli which would include the sounds, the smells, of course, the visual input. It might even be the flavors of the food that you eat. Of course, you better be wearing your mask these days. So all of these come together to give you a memory, a learning event, which includes the market. You can have other kinds of features. For example, all of you will recognize this image because of the visual features it contains. Yeah. So the Taj Mahal is recognizable in visual terms because of the various visual components in this. So this is pattern learning of various kinds. And sequences, which is something I've become very interested in, is not just a sort of static set of patterns, but it is a sequence of patterns. And this is something I've become extremely interested in. For example, you can recognize someone by the way they walk, even if you don't see their face, if you know them well. Yeah? You can recognize music. You can recognize hundreds of tunes by the sequence of notes or the sequence of chords, complicated notes. And this is, I think, a really important aspect of learning, that you put together not just patterns, but you put together how patterns relate to each other in time. So these are all aspects of learning, and we'll have a glimpse of a couple of these in a moment. So you've already seen one kind of learning, which was the associative learning, right? So you have association between the bell and the food enough times, and you strengthen the synapse, and you end up with the capability that the bell will activate the salivation. And this is a form of learning. This is a behavioral outcome of some cellular events, and as we shall see in a bit, some molecular events. And we can actually sort of trace this entire hierarchy of learning right from the molecular to the behavioral level in, maybe not in a dog, but there are certain much simpler animals like slugs, in which, sea slugs, in which this very, very linkage has been established. So this is the association form of learning, what to learn, associations. And this can be done, as I said, through molecular events. So here's an example of one of the key forms of molecular events that does this, right? So we've already talked about presynaptic and postsynaptic cell, right? The presynaptic cell was the bell, and the postsynaptic cell was the food neuron, right? So when the things are idle, if there's no food, let's say there's no food and the bell rings, what happens? Your presynaptic cell is active, it fires, it releases neurotransmitter, but the postsynaptic cell will get that neurotransmitter, but it's not going to have an association. There's no relationship between the bell and the food because there's no food at that time. But supposing you have both the bell and the food at the same time, that's when this particular molecule called the NMDA receptor acts like a molecular AND gate. It does association between the pre and postsynaptic activity. And it's a really very cool process. How it works is the presynaptic terminal, the bell neuron, is active, and therefore it's releasing neurotransmitter. The neurotransmitter is going across, it's flooding the system, it's trying to open the channel. But this channel, the NMDA receptor, won't open. It'll try to open, and it can, but the thing is the NMDA receptor is blocked by an ion of magnesium until the postsynaptic cell, the food cell, is also active. And when the food cell becomes active, its potential becomes large, more positive. The positive repels the magnesium ion, which is positively charged. And now you have the conjunction of the pre and postsynaptic activity. Now this channel opens. Now this molecular gate can go into the on state. And so here you have an AND gate, a logic gate, operating to implement this association between that signal and that signal. And so we've gone all the way down to a molecular mechanism to account for associative learning. And this is, of course, just the first step. It's just the trigger. So now the channel opens, calcium comes in, and many, many things happen, which we will discuss in a little bit. But the point here is that here we actually have enough information to come up with a molecular explanation for how you can have association. And there are other forms of associativity, but this is a particularly nice and simple one. Here's another one, which I've been very interested in. This is not associativity. This is a much more complicated form of learning sequences. So supposing we have a sequence, let's say, of numbers, or let's say of notes, yeah? And let's say there's a bunch of cells which respond to that one, another bunch of cells which responds to number 2, number 3, number 4, number 5. And these are connected in a spatial order on a piece of nerve cell, on a dendrite. Yeah? And what can happen is if the connections are in the same order as the timing, then you can get a stimulus activated here that will release, let's say, a compound, let's not give it a name just yet, which starts to diffuse in both directions. If the next stimulus comes and hits the peak of this diffusing molecule as it goes along, that'll give it a boost. The next one comes, it gives it a boost. Next one comes, it gives it a boost. And so if the signals are in the correct order, you can get a boosted output. And this is actually a fairly important, a fairly major computation that can be done at a molecular scale, because it will allow a single nerve cell, in fact, a small piece of a single nerve cell, to recognize, to learn that a certain sequence of inputs has come in and means something special. And just to show you how this might work, I've made a computer model of this, and without going into the details, the blue trace is the sequential input, and the red trace is the scrambled input, where there's no particular sequential order. And as you will see, it'll play again. The blue stimulus, the sequential stimulus, 1, then 2, then 3, 4, and 5. These are playing out in order. That gave a very large response. And now we can contrast it with the scrambled sequence in red, which will play back again in a moment. So the red one started there, then it went there, then it went there, then it went there, then it went there, completely scrambled. And as you can see, the red response was very, very small compared to the blue response. The simple thing, as I said, is that if you have it in order, then the reactions get to build up on each other. The molecules, it acts like a cascade, and you get a strong response as a result. So this is a molecular mechanism for doing a rather sophisticated calculation, which is to recognize a sequence in time of different kinds of stimuli. And I think, and this is one of my areas of research, that this is absolutely crucial aspect of what to learn, how people learn certain things. Okay, so this was the topic of what to learn. Let's move on. When to learn it? When should you learn something? What kinds of context is good for remembering something? Well, the obvious one is that you should be alert, right? So now it's just before lunch, and so you are all reasonably alert. Your insides are presumably not full of food and not busy digesting stuff. And hopefully it's been a while since you got up. So you're alert. Coffee might give you a little bit of an additional boost to alertness. And this is a very good context in which to learn. Another thing that is important for learning is salience. Is this significant in some way to you, right? So you might not learn, let's say, the last column of the newspaper when it's talking about some event somewhere which you're not at all interested in, but you would probably remember the headline of the paper, especially if it told you that, oh gosh, today the COVID infections are really huge and my area is going to be locked down, right? So salience is also very important for what you learn. And very closely tied up with these is emotion. And this is, again, where one comes back to molecules. So the molecules, the neuromodulators, control many of these aspects of emotion, right? So anger is through certain norepinephrine, adrenaline, all of these things. Fear, the reward signal when you achieve something maybe that you didn't expect, when you get a reward that you didn't expect, or something that you've been working for and finally it gets rewarded, that is very, very important for salience and emotion. That gives you an emotional kick. But equally, negative emotions can be a very strong basis for learning. And this is unfortunate because, for example, people who are exposed to trauma may suffer from post-traumatic stress disorder. They cannot forget the event that led to that learning. So this is why it's not just important to learn, but to also modulate how much you learn. And in some cases, in many cases, it's important that you also be able to forget. For example, post-traumatic stress disorder is a disorder, not because you learned it, but because you cannot forget it. You cannot let go of it. Yeah? And this is one of the reasons why messing with brain chemistry, which is what happens when you take drugs of recreational, it's a bad word, but when you take drugs that you should not take, they mess with your brain chemistry. They may give you reward signals, and they completely mess up the very delicate balance that has been established by evolution over billions of years to make sure that the learning and the forgetting maintain a reasonable balance so that you can continue to function through your 100 years, hopefully, of life where you are able to keep taking in new experiences without being overwhelmed by old ones, right? So don't mess with your brain chemistry is the short point here. But this is all happening through reinforcement signals. And of course, you probably know that dopamine is a very important reinforcement signal. In fact, many drugs of abuse activate the dopaminergic signaling pathway. I'm not going to give you an exam on this, but just to illustrate, we were just looking at the glutamatergic input. This is the one that opened the channels when there was association. And the dopaminergic input, the dopamine signal, comes in and interacts with this so as to modulate how much learning is happening through the associative pathways, right? So the dopamine is a positive enhancer of learning. It's typically thought of as a reward signal. It does many other things in the brain. And this is one of our current understanding of how it does so. The signaling molecules interact with each other such that the learning that is happening here is hugely amplified in the presence of dopamine. And these neurons, these medium spiny neurons of the striatum, they are absolutely crucial to this. As many of you no doubt know, if you lose these neurons, you end up with all sorts of disorders, Parkinson's disease being a major one of these. There's a remarkable, they're a very small population of neurons, but they have an incredibly huge effect on brain function. So this is how this is done. Okay. All right. There we go. All right, so now let's go to the next section, which is how do you store this information? We've now figured out what to learn. We've learned associations or sequences. We figured out what tells us when to do this learning. Now let's talk about storing the information. Now you're all familiar with this. This is now starting to get to be old technology, a hard disk. And the reason why this stores information is that you make changes in it, physical changes, magnetization, and those changes last for a very, very long time. And the sort of old-fashioned idea about the nervous system was that, yeah, you make structural changes to these synapses, and once you made them, they just stay there. They last for a long time. Well, that's not actually true, because as I mentioned, typical proteins last for at most a day or so in your brain. So that won't work. Here's another kind of memory. DRAM. And the way that this would work is that you store some bit of information, and then you need to periodically refresh that information. If you don't refresh it, it forgets it. And there is a brain analogy of this. That is, you can have a little circuit, it's called a reverberatory circuit, where, say in this case, 3 example neurons are mutually activating each other. And this is thought to possibly play a role in something like short-term memory, for example, trying to memorize a phone number, which you forget as soon as you don't need it. And so this might be one way to store this information. That is, you activate that goes there, that goes there, that goes there. And so they successively activate each other. They refresh each other over time. This is a form of memory which many of the IIT students will be totally familiar with. This is a bistable gate, a bistable system formed out of 2 NAND gates. I won't go into the details of this, but it has, as long as power is provided, it will store information of its past state, depending on the inputs that it is given. And here the equivalent, and the key thing here is there's a feedback loop here. And there are many, many feedback loops in these chemical networks. And I'm going to make the point that that kind of feedback works for storing information in the brain as well. So I don't know how many of you recognize this picture. This is a great surrealistic painting by Salvador Dali. It's called Persistence of Memory. And I like it because it actually, it's counter to one of the big problems in studying the brain, which is that these spines, these connections between cells, are anything but persistent. They are at face value a very bad place to store information, right? So the reason I say this is that, first of all, I already mentioned proteins or other molecules in the brain, they last for minutes to days. So how are you going to store information for a century? There's another big problem, which is stochasticity. There's a huge amount of chemical noise, reaction noise when you scale things down to this tiny volume, when there's maybe 5 free calcium ions, 5, count them, 5. Right? And then diffusion simply flushes things back and forth so rapidly that how are you going to store anything? How are you going to keep any chemical gradient there at all? So I'm not going to address all of these questions, but maybe I'll address this one a little bit, which is the turnover problem. How do you store information even when the molecules keep coming and going? And the way we do this is through feedback, the bistability thing I told you about. And the way it would work is, supposing you have a molecule A that activates molecule B, and molecule B that in turn activates molecule A. So at resting state, they're both off. You give them a strong stimulus, and now they're both active, and they mutually turn each other on. Of course, it's not a test tube full of molecules. It's individual small numbers of molecules. And so that's what I'm representing here. Yeah. So you give a strong stimulus, and it changes, the whole system changes into a different state. And this remains. And just to reiterate, this is a bistable system where you have 2 states. You can think of them as energy minima, and there's some kind of unstable state in between them. And if you push hard enough, you can go from this state across the unstable state into the other one. And those of you who are a little bit familiar with this kind of analysis, you can make a phase plot of this. You can draw the null lines by examining the effect of B upon A, which you can draw as a very simple chemical dose response curve. Yeah, all chemists will recognize this kind of typical reaction shape. You can do it the other way, the effect of A upon B. And now so these are both steady state curves. And now if you plot them both on the same axis, which you can do by sort of rotating this curve along the 45-degree line, so that the axis is both A and B, you'll find that there are intersection points of these plots. And these intersection points are really important because they define the steady states of the system. And typically you will get just one intersection point, like so. But in specific configurations of feedback, you can get multiple intersection points, one there, and then one intermediate one. And it mathematically turns out that these are identical to state 1, stable, state 2, stable, and the middle point being the unstable intermediate point, right? So this is mathematically how you can show that chemical systems through feedback can achieve storage of information. Okay, fine. So you can store information. How do you ensure that you can survive turnover? And this is how it would work. So these sort of greenish gray clouds are meant to represent the dendritic spines, the little regions where the synaptic input comes in. And the crosses are meant to indicate turnover. The molecules have been washed away. They have degraded. And if this were to continue, very soon you wouldn't have any molecules left at all. However, the cell is alive. Therefore, it's making new molecules all the time. These come in. But the trouble is that these molecules do not know the current state of the system. They're naive. Fortunately, the thing about bistable systems is that a small perturbation, such as a bunch of new naive molecules, does not change the state. It will restore itself. So these new molecules are sort of, so to speak, they're entrained to take the same state as the other molecules. If it was active here, they too would become active. And now they too sustain and support the memory. And in fact, this is not too different from, let's say, oral traditions in learning the epics. You know, the guru might die, but the disciples have learned what he taught or she taught. And so over generations, the information is preserved, even though the molecules, in this case, the people learning it, may pass on. So this is how you can store information beyond the lifetime of the molecules. And this is something that I've spent many years studying. It's very, very interesting to see how it works. Okay, and so now just to bring this back into context, think of this little feedback loop here in the synapse being the point at which you're storing the information. That is, the chemical reactions that allow for sustained activation are what are maintaining the strength of the synapse. Yeah, so if the synapse has a lot of this correct kind of chemical activity, which might, for example, be increasing the number of receptors, then that synapse will be strong. It will mean that the bell will have a large effect on the food neuron. Yeah. So, and you can flip the state from the active state to the inactive state, depending on the kinds of chemical signals you give it. So this is relating chemistry, the feedback system of chemistry, back to our toy model of how you could remember something in Pavlovian conditioning. So we talked about associativity, and now we've talked about how you store the information once the association has been made. So finally, how to recall it. I'm not going to belabor this point because you are all probably much more familiar than I am with the vast literature on neural networks and AI and so on. Suffice it to say that there's many ways you can do this. We still don't really understand how the brain does it, but it has probably got some ingredients of these kinds of networks. So for example, here's a typical feed-forward artificial neural network. And with 3 layers, a hidden layer, sorry, an input layer, hidden layer, and output layer. And as all of you know who have looked at this kind of thing, the key thing is, what are the synaptic weights? What are the connection strengths? And remember, we are storing connection strengths through these feedback loops, through the molecules in the brain. So you can very easily imagine that here's a network of neurons. They have the connections. Some of the strengths are large, some are small. And by the information flow through these networks with these weights, you're able to store information and recall information. For example, this old paper by Terry Sejnowski trained this neural network to pronounce English from spelling, which is, of course, as you all know, a terrible challenge because English spelling is anything but logic. Neuroscience is spelt like that, and it goes through this network and it comes out spelt pronounced correctly with the correct phonemes, neuroscience. Though, anyway, you can try and imagine how a naive person might try to say this. Okay, so this is one form in which these chemical storage of information is being read out into the behavior of the system. Another kind is an associative memory, so where you have feedback in the system. And this is, again, very, very common in different kinds of neural networks. This kind of network can do things like pattern recognition and completion. So if you store 2 different patterns, a face-like pattern and some kind of random pattern, then if you give it only a partial stimulus, over a couple of cycles, it will recreate the full stimulus. It will remember the full pattern. If you give it a pattern which is even slightly like this, it can also recollect it, right? So, and there's lots of theorems about the capacity and so on, which I won't go into. But the point I'm making is that all of these depend on synaptic weights. It is the connection strengths, which, as we discussed, can be thought of in terms of the chemistry. You all know about deep learning. You know, perhaps the one additional thing which is brought in from brain science is the point I made about motivation, about salience, the dopaminergic signal, right? Reinforcement. There's a whole field of reinforcement learning, and that is in its ancestry, that is derived from concepts about the reinforcement chemicals that the brain secretes in order to decide whether or not something should be stored. And nowadays, you can do amazing things with these networks. So I'm going to now begin to wrap up, and I'm going to take a much more general philosophical point, talking about curiosity, learning, and survival. So curiosity, even though there's the saying, right, curiosity killed the cat, but actually, no, curiosity is a hugely important, it's a critical survival trait. Almost all animals will deliberately go out to explore their environment. They will deliberately go out to seek information. And it makes total sense. The more you know about your environment, the more you know about social environment or physical environment or biological environment, the more likely you are to make survival favorable decisions, right? If you're a rat or a mouse and you don't explore your environment, you just go out, you're going to be eaten by the cat. But if you sniff around a bit, explore it a bit, and you smell, aha, there was a cat there, and choose a different route, then you are more likely to survive. Exploration is key. As we go to more intelligent animals, there's a certain level of understanding one's environment that one builds up. And by understanding it, you know the cause and effect, the associations, the sequences in which things happen. Animals can learn to do much more sophisticated things. And sort of a culmination of that is science, where we are engineers and scientists. We explicitly study these really complicated relationships in the natural world to better understand it. And this is what gives us the incredible capacity to support 7 billion plus human beings on this planet in hopefully steadily increasing well-being. So this is made possible because of curiosity. It has allowed us to survive. Here's another important survival trait, which, it's kind of, I find it kind of surprising and depressing that so many political movements seem to go the other way. Diversity is a hugely important survival trait. This is shown time and time again in the fossil record. That is, the more diverse genera is, the more diverse a set of animals are, the more likely it is that some of them will be in the right place with the right characteristics to survive. So that's a simple form of genetic diversity, and that humanity has a certain amount of genetic diversity. Many animals have much greater genetic diversity. And that is important for survival. If you have a monogenic culture, a monogenic society, a disease, for example, that goes after those traits is just going to wipe out that population. Diversity in genetics is extremely important. So what does this have to do for learning? The point of this is that humans have, by and large, shaped their environment because of their capacity to learn. And this is done in a social context. And the social diversity is also an incredible resource for humans to be able to handle different situations, to be able to deal with the incredible scaling up of human civilization, because you have to meet so many different kinds of people around the world. You must be exposed to this kind of diversity in order to function effectively. And that's not the only thing. In the case of learning, there's a specific and very well studied process, very well studied property of learning of the brain, which is called the enriched environment thing. And let me just give you an illustration of this. Many aspects of healthy brain functioning are related to an enriched environment. So for example, if you put a mouse or a rat in a nice busy cage with lots of friends, a running wheel, ladders and things to climb around, pipes to run around in, and you compare that with a mouse or rat that's in the standard environment, which basically has the food and water and nothing else, yeah? And you look at the neurons, and the neurons in these animals are much more flourishing. You can see them, they're branching more, they have more connections, they last better, they are able to withstand stress, they're able to do many things far more effectively than the neurons that are present in animals in a standard environment. And I think this is extremely important, I think, for all of you young people out there. Yeah, you need to make it a point to hear views that challenge your views of the world, to seek out diversity of opinion, of ways of doing things, of personalities, of social structures because this is going to enrich you. This is what makes it possible for you to function as a human being in a complex world, but it also enriches you in terms of your own health, in terms of your neural capabilities. So it happens for rats and it happens for humans too. Be enriched. Don't just hunker down in your dorm room and do nothing except study your course. There's a lot of amazing things in this world, and you should enjoy them, and it's good for you. So let me now summarize. I've gone through talking about the brain and metaphors. And for example, as I said, I think one of the key metaphors is the brain as a computing unit. And there are more modern metaphors. The AI is sort of, I said, an inverse metaphor for the brain. I gave you a sketch of how learning happens through this toy model, this cartoon version of a dog, which learned Pavlov's classical conditioning. Then we went through the different operations in learning, what to learn, associations, for example, when to learn it. For example, when there's a signal saying that this is a rewarded experience, the dopamine effect, reinforcement, how to store information. There we discussed feedback loops and bistability and how this can be implemented through the chemistry, and how to recall it through the various neural network architectures, and probably many, many more that the brain is doing that we are not yet aware of. And so finally, I sort of touched on a little bit more philosophical things, saying that, you have this incredibly marvelous machine in your head, and it benefits from learning. It's good for your health. It's good for you. And above all else, it's actually a lot of fun to learn. And I'm sure that's why you're here today. But I really urge you to go out and make the most of it. I'll give you a quote, which I think exemplifies this. It's a quote from a poem by Tennyson. He says, "All experience is an arch wherethrough gleams that untraveled world whose margin fades forever and forever when I move. There's always more to learn." So I'll leave you with this thought. You have a brain, use it. Thank you.
Thank you, sir, for such a beautiful and enriching session. I hope the audience will post their question in our comment section. And I request my friends to ask the question that they would like to ask now.
Thank you, sir, for such an insightful session. Arnav here. Sir, we have got a few questions from YouTube comment section. So one has asked, could you please suggest some of the most exhaustive open source simulators?
Sorry, can you speak up a bit? I can't quite hear.
Could you please suggest some of the most exhaustive open source simulators for modeling single neurons and biological neural networks? Also, also at the moment, what is the best way to model neural networks with electrical gap junctions in addition to chemical synapses?
So the most popularly used simulator for biophysical detail is a simulator called Neuron. That's been used, for example, in the Blue Brain project and many other projects. We have our own simulator called Moose. Moose does that kind of simulation, but since we do a lot of chemical simulations, that is one of the emphases that we have, multi-scale models, which include both electrical and chemical signaling. If you want a little bit less detail, there's a lovely simulator called Brian, which is very effective if you want to do small networks. So all of these simulators, well, maybe not Brian, but certainly Moose and Neuron will happily do gap junctions and other kinds of networks, other kinds of synapses.
Thank you, sir. So the next question is, we have heard that an average human being can tap into around 5% of the brain's total potential. Is there a way in which we can tap into more than that 5% to help enhance learning?
Okay, that's pop psychology. That's not a correct statement. Yeah, so we actually use a lot of our brain a lot of the time. The point is, which I'm trying to emphasize towards the end, is that the richness of your experience base, of your database, of your scope for thinking about the world comes from how much you have learned about many, many diverse things. So the way, if you want to, I mean, you're already using all of your brain, but if you want to use it more effectively, then Broaden your experiences. Yeah? Listen to talks, read books about subjects you never thought about. Hear viewpoints that you may not agree with or that you never even thought you'd be interested in. Go places. Yeah, do that, and that is a good way to use your brain more effectively.
Sir, I will add one more question.
Yes.
How do electrical gap junctions configure and store connectivity strength?
So electrical gap junctions are in the human brain a somewhat specialized feature. Their main point is that they are so let me step back a bit. As their name indicates, they are connections made sort of cell to cell, which allow electrical currents to flow directly between the cells. And it's called a gap junction because of the nature of the proteins that make a little gap, make a little channel, and there's a small gap between the membranes as it does so. Yeah, so that's fine. It's fast, but it's got one incredibly big drawback or limitation, which is that in a chemical synapse, you can change the sign, you can modulate it, and you can learn. Right? So actually, those are three things. But these are all things which are attributes of chemical synapses, not of gap junctions. And the reason is that the chemical synapses, which are incredibly complicated cell biological processes, they allow for all of this chemical signaling that we discussed. All of the events that I discussed happen through chemical synapses. And that is why, even though gap junctions are faster, because they don't have so much cell biology in them, so many mechanisms to take place, they are not as versatile. And for the brain, versatility is a big deal.
So Arnab, do you have any other question to add up?
Yeah, one last question. From the knowledge out of your lab and others, is memory a neural network or cellular phenomenon? We have got
the answer is yes. It's all of the above. I mean, I think one of the slides I had, and I emphasize this point, is that the brain does computation at many, many levels. It does it molecular, synaptic, subcellular, cellular, circuit, network, area, region, and so on. It does it at all of these levels. And these are all very, very tightly interlinked. You cannot, if you just study one of them, you're going to miss the reality of what's going on. So memory is a function of all of these levels. There's certain aspects of it, such as connection strengths, which are substantially managed at the molecular level. But there's genetic input into that, there's electrical input into that, there's network input into that. So you cannot reduce it beyond a point. You really do have to remember the context, and the context is that the brain is doing these things across all scales.
So thank you, sir, for such a beautiful and enriching session. I hope the audience has thoroughly enjoyed it, and I request you to kindly go through our videos, and the upcoming sessions will be by 2 p.m. So please join us by 2 p.m. I request everybody. So have a great lunch.
Yep,
and thank you, sir.
Thank you very
Thank you
much
for joining
to my
with
audience,
us, sir.
and thank you very much to the organizers.